Hello everyone, and welcome to the last lecture of Unit 2. This lecture covers the material in Chapter 11, and this is a relatively short chapter and relatively short lecture. It discusses some of the ways that microbes can interact with each other and how those interactions can either be helpful or harmful to the organisms involved. And this information about those types of interactions actually applies to the interactions that we have with microbes as well. As always, you have your lesson objectives here. Feel free to use those in whatever way is most beneficial to you in terms of guiding your note-taking and listening for key information. Although we don't always talk about this or focus on this, it's important to understand that most of the time, microbes do not exist solely by themselves. So even in situations where we're talking about one specific type of microbe being able to cause a certain disease, rarely would you find a situation where that microbe is the only one present in that given environment. Most of the time, microbes are constantly interacting with other types of organisms, whether those other types of organisms are people, other microbes, plants, animals, etc. So rarely do we have situations where we can truly say there is just one type of microbe in a particular scenario or situation. And if we can say that, most of the time we're talking about a lab setting. The types of interactions that microbes have with each other and other types of organisms can vary. Sometimes these interactions are beneficial, and sometimes they are kind of neutral, while other times they can be very harmful. We can group the types of interactions microbes have with other microbes and things like people, plants, and animals into two very broad categories. Those categories are symbiotic relationships and non-symbiotic relationships. Now, this is another kind of tricky piece of vocabulary because oftentimes this term is not actually used the way it's supposed to be used. So if you read something about symbiotic or symbiosis in the media, like on social media or even in the newspaper, oftentimes the way that's being used is incorrect. So this is another one to kind of pay a little bit more attention to so that you make sure that you are fully understanding the actual definition of these terms rather than the way the terms get used in everyday language. A symbiotic relationship is one in which two or more organisms interact, and at least one of those organisms must have that relationship or they will die. A non-symbiotic relationship is a relationship in which two or more microbes interact, but they don't have to have that relationship to survive. The microbes that are involved can live on their own just fine. Within the category of symbiotic relationships, there are three main varieties. There's mutualism, commensalism, and parasitism. With mutualism, this is a relationship where two or more organisms interact and both of them benefit. Additionally, at least one of them requires that relationship to stay alive. One example that I have is the relationship between termites, protozoa, and bacteria. We've talked about this a little bit before, back in Unit 1, but termites are insects that eat wood. However, the termites cannot actually digest the wood themselves. They rely on the protozoa and the bacteria, both types of microbes, that live in their intestines to produce enzymes that will actually break down the wood or the cellulose that's in the wood. If the termite did not have the protozoa or the bacteria, it would die because it would not be able to get energy from its food source. It would not be able to produce ATP, and that would be it for it. So in this relationship, all three of these organisms benefit from this relationship. They all get something good from the relationship. However, at an absolute minimum, the termite has to have this relationship or it will die. The protozoa and the bacteria might be able to live on their own, but In this case, they're better off with the termite. The termite, though, absolutely must have the protozoa and the bacteria, or it will die. In commensalism, there is an interaction where two or more organisms are interacting. One of them benefits and one of them is unaffected. There are not a lot of great examples for this one, but there is one that happens inside the human intestine between two bacteria called E. coli and Bacteroides. So E. coli are facultative anaerobes, meaning they typically prefer to use oxygen if it's available, but they don't have to use oxygen. They can use something else to make their ATP. Now they live in the intestine and kind of just coat the surface of our intestine. Those red lines are our intestine, and these are E. coli. So E. coli are sitting here growing in our intestine, and they are very happy. They can use oxygen to make their ATP or not. It doesn't really matter to them. Either way is fine. But this other type of bacteria, Bacteroides, is an obligate anaerobe. It cannot live with oxygen, and it absolutely will die if oxygen is there. So these two organisms interact inside the intestine where E. coli is able to take up the oxygen and create these little pockets of anaerobic environments. So these ones I'm drawing in green, I'm going to see if I can zoom in. Not great. Okay, hopefully I can zoom back out again without distorting everything. Basically, the E. coli here are in blue, and they will use oxygen if it's available, so that's good for them because it helps them make more ATP than other types of electron acceptors. And by using this oxygen, they create these little pockets where there's no oxygen left. And that allows the Bacteroides to kind of grow underneath them or near them, associated with them in some way or another. So in this situation, the Bacteroides absolutely must have this interaction. They live with the E. coli and they have to have it or else they will die. The E. coli are not affected by this other type of bacteria at all. They will grow just fine without it. They will grow fine with it. It doesn't matter to them at all whether this other type of bacteria is there or not. So that's an example of commensalism. Our last type of symbiotic relationship is parasitism. In this type of relationship, one organism benefits and one is harmed. We can see this type of interaction with basically any type of infection. If we are infected with, let's say, COVID or tuberculosis or staph, those organisms are benefiting. So they have this relationship with us, and at least in this scenario, they have to have that relationship to stay alive. They are benefiting from being able to live inside our bodies, but we are being harmed. So I created this table for you. Unfortunately, there aren't a lot of great pictures for these types of concepts, but you can use this table to understand how the relationships work in terms of who is benefited and who is harmed. So with mutualism, remember, this is a relationship where two or more organisms interact. At least one of them must have this relationship to be able to survive, and all of them or both of them benefit. With commensalism, the organisms interact. At least one of them must have the relationship to survive. One of them benefits, and the other one isn't affected at all. Doesn't matter to them either way. With parasitism, two or more organisms interact. One of them benefits, one of them is harmed, and the parasite, the one that's doing the harm, has to have the relationship to survive. Non-symbiotic relationships are those in which two or more organisms interact and neither one of them or none of them need the relationship to stay alive or to survive. There are two types of non-symbiotic relationships. One is synergism. In synergism, both of the organisms benefit from the relationship, but they don't have to have it to stay alive. There are actually not a ton of examples of this that we know of in the microbial world, although we are discovering more. But there is one really intriguing one, or at least one that I think is really intriguing, between two species of bacteria that commonly are found in the soil. So in this scenario, the first species is what we call Arthobacter, and the second one is what we call Streptomyces. And these are just very, very common soil bacteria. They live in the soil, they gather nutrients from the soil, they get their carbon, their electron acceptors, all of their nutrients, everything's good, they survive, no problems. However, an interesting thing happens when you put Arthobacter and Streptomyces near each other in the soil. When they are together in the soil, each of them produces 1/2 of an enzyme that they are able to secrete from their cells, and when it's in the soil, those 2/2 of the enzyme actually join to make a whole enzyme. That enzyme is able to break down certain types of compounds in the environment, especially things like pesticides and insecticides. And then each of those bacteria is able to take in those broken down pesticides or insecticides into their bodies and use them as a source of fuel to make ATP. So they can use them as electron donors to create ATP. Alone, each of them only makes half of this enzyme, and the enzyme doesn't work. But together, they each make half of it, and then they secrete it. It becomes a whole enzyme, and it's an exoenzyme in this case. And that exoenzyme is able to break down these substances in the environment that the bacteria are then able to use as another source of electron donors or nutrients. So that's a pretty cool thing that alone they're doing fine, but together they're better off. The second type of non-symbiotic relationship is antagonism. Antagonism occurs when one benefits and one is harmed. So these two organisms interact and one benefits from it, but the other one gets hurt from it. A great example with this is actually through antibiotic production. So these days, most antibiotics are produced in laboratory settings or in clinical settings, and they are created in the lab, they're synthesized there, or at least partially synthesized there. But antibiotics are technically natural compounds that are produced by certain species of fungi and certain species of bacteria to kill off other ones near them that might try to invade their space or take food or something like that. So many fungi and bacteria that live out in the environment produce these chemicals that we call antibiotics and secrete them out into the soil or out into the environment. And those chemicals kill off nearby bacteria. In this situation, the one that's benefiting is the one that is producing that chemical, either that bacteria or that fungus that is producing the antibiotic. And the one that's being harmed is obviously the one that is getting killed by it. The one that produces this antibiotic is benefiting because it's killing everything around it, or killing many things around it at least. And that means it has more space to grow, it has more nutrients available to it, and it itself won't get attacked by something. So that's a great example of antagonism out in the environment that we've actually been able to take advantage of, because these antibiotics are natural compounds that are produced by many different types of bacteria and fungi out there as a means of kind of self-defense or self-protection. And we're able to benefit from that by forcing them to produce those antibiotics, and then we can harvest them and put them into pills or IVs, etc. One last topic I want to touch on in this lecture is the concept of biofilms. We've talked about them a little bit before, but now I want to go into some more detail. Biofilms are special microbial associations. Technically, they can have multiple types of interactions going on within them. So within a biofilm, you might find organisms that are in symbiotic relationships or non-symbiotic relationships, and those could be something like mutualism or synergism or commensalism or antagonism even in some cases. But basically, Biofilms are this special type of association that many microbes can create. And at their basic level, what they are is a bunch of individual microbes that have kind of joined forces, that have combined their capsules or their slime layers together, whatever their type of glycocalyx was, and they grow together in this structure that's called the matrix. So the matrix is essentially a bunch of glycocalyces joined together. It's primarily made of things like polysaccharides. There are going to be proteins in there. There might be some DNA and nucleotides floating around as well. And these associations allow microbes to basically behave as one unit instead of just as individual cells. So the way biofilms form is that you have individual cells. These are called planktonic bacteria because they're acting as plankton. They're just kind of floating around in a surface. So planktonic cells are those that are just floating around in the environment. And these planktonic cells attach to something. They find some kind of surface in their environment and they settle down. They form an attachment onto this surface. When there is a sufficient number of cells that are living together on this surface, a quorum is formed. Now, a quorum is the number of cells that is required to start a biofilm. So the number of cells to start a biofilm varies. There's not just one number I can give you. It depends on the type of cells we're talking about, depends on the type of environment, depends on potentially nutritional factors, pH, stuff like that. But whatever that exact number of cells is, once that is reached, a quorum is formed. When the quorum is formed, the matrix begins to develop, and the matrix engulfs and kind of protects all of these individual cells that are living within it. As the matrix matures, the cells begin to coordinate their activities and act as a single unit with a single goal, basically. Now remember, these are not conscious organisms. They're not doing this intentionally. It's just that all of the metabolic reactions in these cells start working together. Even though these are individual organisms, they all start coordinating their activities, working together, and this allows them to function as a unit instead of individuals. Oftentimes, as the biofilm grows and gets larger and larger and larger, pieces of it will break off and migrate to a new location. This is great for them and less great for us if we're talking about this happening in our body, because that means the biofilm is spreading. One of the things that is particularly significant about biofilms is that because the cells are working as a unit rather than individuals, they are very, very difficult to disrupt, almost impossible to get rid of, and oftentimes antibiotics that would work on the planktonic cells, when the bacteria are just individual cells, do not work when they're in the biofilm formation or when they're in that matrix. There are various reasons for the antibiotics not working. Sometimes it's because the antibiotics can't penetrate the biofilm. Basically, you can kind of get the antibiotics to the top few cells, but there will always be ones at the bottom that can kind of regenerate. Sometimes it's because the close proximity of cells allows them to go through things like conjugation and transformation very quickly, so they can pass off the DNA that's required for antibiotic resistance very easily. And other times it's more of kind of a concentration issue. Because they are all growing together, they survive the immune attacks better and they increase in numbers faster than we're able to dose the antibiotic in terms of providing the appropriate concentration to get rid of them all. So there's lots of different reasons why antibiotics might not work in biofilm situations, but they do cause all kinds of problems once they are in the human body or the animal body and they form there and get stuck there. Although many different microbes can be involved in biofilms and are involved in biofilms, there are some very common ones that tend to form biofilms in the human body. So the specific microbes that are kind of most common to form biofilms in the human body are those of the Pseudomonas species, the Streptococcus species, the Staphylococcus species, and E. coli. You don't need to memorize these for now. I just like to mention them because sometimes people do have questions about these, or they have experience with it. Either they have had a biofilm infection or someone they know has had a biofilm infection, and it helps them kind of put this into context. In terms of the problems with biofilms, we talked about that a little bit on the previous slide, but essentially once microbes are in a biofilm, they have different metabolic characteristics, and as a result, they oftentimes are resistant to antibiotics. They're very difficult for us to get rid of, and it makes it so that once they get started in the human body, they are stuck there unless some kind of extraordinary measure is taken. In terms of treatment for biofilms, oftentimes that requires multiple different types of antibiotics for long periods of time. In the United States, a typical course of antibiotics is around 10 days. That can vary depending on the situation, the prescribing healthcare provider, and various other factors. But 10 days is kind of a standard course of antibiotics for many types of infections. However, if someone has a biofilm, they could potentially be on antibiotics for months or even years, because there is just no way to truly get rid of this structure that has formed inside the human body. And antibiotics are used to control the infection, but not necessarily eliminate it. In the last couple of decades, as these types of infections have become more prominent, or at least more people are aware of the fact that they exist, various types of equipment has been produced to help monitor these types of infections. There are special devices that allow healthcare providers to look inside places that oftentimes form biofilms, places like the lungs, the urinary tract, the ear canal, and kind of monitor the progression of that biofilm. There's a really cool scope that's used in the ears, especially for children who get biofilms that form in the ears, that the physician or other healthcare provider can insert into the ear and just look at the level of that biofilm kind of over time and get an idea of whether the treatment they're using is working or not and whether they need to switch it. Ideally, in the future, we will be able to regulate these biofilms with different types of treatments. Our normal antibiotics, or at least the standard ones that we have now, work in ways such as trying to stop protein production or interfere with the cell wall and things like that. And that is great for many types of infections, but not as great for biofilms. It just doesn't work as well. So there is a lot of research going on right now to try to figure out if we can create a medication that will interfere with the genes that are used to actually make the biofilms. So if we can interfere with the genes that the microbes are using to make the biofilm and kind of keep up the biofilm, then we can basically stop the biofilm from existing and treat the infection with existing antibiotics. Because what it would kind of be doing is turning back time and taking these microbes from a biofilm state back to the normal individual planktonic bacteria, and then they will be sensitive to the antibiotics that we already have, that already exist. So we don't necessarily need to provide months or years long treatment with antibiotics or even develop new special antibiotics for biofilms. We just need to be able to stop them from creating the structure, and then we can treat them as we normally would.